Good morning. My name is Steve Minnie, and I'm the Senior Director of Applications for the AFM Business Unit in Bruker, and I'd like to welcome you to this installment of the Bruker AFM webinar. The presentation is titled AFM Enhancing Traditional Electron Microscopy Applications, presented by Johannes Kent. Before I get started, though, I'd like to first, we encourage your participation during this webinar, and if you have a question, please type it into the questions dialog box on your screen. We'll accumulate these questions through the presentation and group them together and answer them at the end of the prepared remarks. Quite often we have more questions than we can answer in the session, and in those cases we'll be sure to follow up with the questions afterwards. Also, if you'd like to review the presentation afterwards or pass it on to a colleague, the webinar will be posted online, most likely later today. And it's located in the webinar section of the Bruker webpage. But also as a follow-up to this presentation, you'll be receiving an email with the link. Finally, when you exit the webinar software, we'll ask you to take a brief survey, and we'd very much appreciate it if you could take the time to complete this, as it really helps us pick the topics that are most important to you and generally make the webinar series better. So let's get started, and let me introduce our presenter, Dr. Johannes Kent. Johannes received his undergraduate degree in physics from the Technical University at Kaiserslautern, Germany. For his graduate work, he joined the laboratory of Paul Hansma at UC Santa Barbara on a Fulbright scholarship and received his PhD for new designs for high-speed AFM scanners and his research on the ultrastructure and nanoscale failure of bone and other biocomposites. He joined Bruker, the AFM development team, in 2005 and has led development efforts on a number of projects, including high-speed AFM technology and the AFM integration, high-performance optical microscopy, and with cell biological methods. His current focus is on the development of high-speed AFM applications, cross-technique high-resolution microscopy, and the adoption of the AFM into new areas of research. Johannes is an author of over 25 peer-reviewed publications and holds several patents. He currently lives near Mannheim, Germany. Now, let me turn it over to Johannes for his talk entitled AFM Enhancing Traditional Electron Microscopy Applications.
Hello and welcome to our today's webinar about recent advances in atomic force microscopy, which we at Bruker expect will start to bring the unique strengths of AFM to some research environments that rely on the high day-to-day productivity and performance of electron microscopy techniques. After over a decade in AFM research and technology development, I am quite excited to see the AFM take its place between the established high-resolution microscopy techniques. This is driven by the AFM's traditional strengths of high-resolution imaging and 3D metrology in different environments, combined with recent advances in quantitative mechanical property mapping, imaging speed, and ease of operation. First, a quick background. Unlike light microscopy or electron microscopy, which use a focused beam of photons or electrons to interact with an image sample, the AFM interacts with the sample surface through mechanical contact. An ultrasharp tip is in typically intermittent contact with the sample surface. The tip is on the end of a micromachine cantilever, and the deflection of the cantilever is observed, in most cases through a laser beam that is reflected off the back of the cantilever and into a segmented photodiode. To form an image, the tip is raster scanned relative to the sample, and line by line a topography image of the surface is constructed, analog to, for example, a scanning electron microscope or a scanning transmission electron microscope. Since the AFM doesn't use a beam, a physical relative motion of tip and sample is needed. To maintain a constant interaction between tip and sample, the AFM operates in a feedback loop, where a third actuator in the vertical or Z direction acts to maintain the tip-sample interaction at a constant level. This is important to ensure that all parts of the surface are imaged at the same optimal, gentle interaction force. Since the imaging interaction is mechanical in nature, information beyond topography can be gained. More about this in a minute. Unlike other imaging techniques beyond the optical resolution limit, AFM can operate in ambient, most gases and fluids, including many reagents. A conductive surface is not required. Since its invention in 1986 by Binnig and Quaid, about 18,000 AFMs have been installed worldwide, and of these, about 10,000 by Bruker AFM, which was founded as Digital Instruments and was previously owned by Vico. When we compare AFM to electron microscopy techniques, we see that each has certain unique strengths. The SEM's probably most unique strengths are its extreme zoom range from millimeters to nanometers and its corresponding extreme field depth, which enable it to explore very rough samples easily. The TEM holds the record for lateral resolution on most samples, which can be suitably prepped, and sample prep methods are highly evolved. The AFM, on the other hand, holds the record on vertical resolution, often requires minimal sample prep, and permits imaging in many chemical environments. We will see later what that can mean in experimental practice. Various additional techniques exist to obtain more specific contrast between different materials. For the electron microscope, the physical characteristics are mostly based on density and elemental ID. For the AFM, they are mechanical properties such as Young's modulus, adhesion, dissipation, etc. One thing that will jump to the electron microscopist's attention is the huge range over 2 orders of magnitude of AFM lateral resolution. Milestones in EM technology were typically marked by achieving a certain resolution. Why is this not as simple for AFM? AFM vertical resolution is easy to understand. It is set by the vertical noise contributions and is an instrument characteristic. Typical values on a good AFM are between below 0.5 angstrom and can sometimes reach 10 picometers in the right lab environment. Even early AFMs have achieved similar performances. However, the most recent designs combine this performance level with large scan ranges, direct Z-sensor metrology, and large sample and multi-sample automation capabilities, which is much more challenging. Lateral AFM resolution, on the other hand, is more complicated. AFM is a contact measurement. Therefore, the lateral resolution is limited by the area of the tip sample contact. Typical AFM tip radii can be around 2 nanometers. Provided the AFM is well designed and can preserve this tip sharpness while approaching the tip to the surface. The contact area now strongly depends on the stiffness of the sample material, which can range over more than 4 orders of magnitude, from, for example, a mineral or metal on the one end to gels, proteins, and even live cells on the other hand. Parts of this variation can be compensated by selecting a suitably soft cantilever. Like with other microscopy techniques, when pushing AFM lateral resolution, skillfully controlling sample factors, for example surface moisture or ionic screening layers, can greatly improve the final result. Here, 2 examples of resolution, one on a relatively hard material, the other on a soft material, both obtained with a dimension fast scan. This slide shows the surface of Muscovite mica, imaged in air. The left image shows a relatively large scan area, 20 micron of a mica surface previously etched with hydrofluoric acid. The morphology shows multiple edge pits of several microns. The reason for this morphology that can be seen when zooming out like we have done here is that HF doesn't really etch mica well unless there is a local flaw in the lattice. According to the literature, these flaws through multiple atomic layers of mica were caused by natural radioactivity, and their density in mica from different sources is related to local isotope concentrations. The 2 micron zoom in of one of the pits shows the individual mica layers, which are vertically separated by about 5 angstrom. For those familiar with AFM, the imaging times of 13 seconds for the large scan and 5 seconds for the zoom in will be lightning fast. On the right-hand side, we see again a mica surface, in this case without etching, and only a scan area of 20 nanometers. We can clearly see the mica lattice. The AFM expert will find the imaging time of 150 seconds extremely long for this kind of image, which is typically taken 10 times faster even on standard equipment, to counteract instrument drift. The point we wanted to make with this slow scan is how stable the fast scans platform must be in order to image a 20 nanometer area in over 2.5 minutes without any visible drift-induced distortion of the image. This is a high-resolution example on the very soft end of the spectrum, a 2D lattice of protein complexes suspended on a mica surface in buffer solution. A soft cantilever was chosen for this experiment. The 2D FFT analysis gives a good indication of resolution. The hexagonal lattice is clearly visible with the second order at 3.17 nanometers and sharply defined maxima in the spectrum. We will look at some more interesting detail of this measurement later. After this brief technical overview, I would now like to introduce you to some exciting recent developments in AFM technology and review with you how they make the AFM interesting as a complementary microscopy technique in research environments that have been counting on the well-established productivity of electron microscopy. The first AFM were operated in contact mode, in which the AFM tip simply slides across the sample surface while a set tip sample force is maintained. Most of today's AFM are operated in tapping mode, in which the AFM cantilever is driven into oscillation by a small actuator and the dampening of the cantilever by the surface is used for feedback. This is advantageous as it reduces lateral tip sample forces and provides some material contrast by displaying the variations of the dampening-induced phase shift of the oscillation. However, tapping mode has some drawbacks, as the dynamics of the cantilever comes into play and the tip sample force is not directly controllable anymore. Bruker recently released peak force tapping, a new imaging mode based on individual force versus distance measurement for each pixel. While the fundamental idea of using force curves for imaging is not new, the implementation of peak force tapping brought the rate of force curves up from a single curve per second up to 8,000 curves per second, while maintaining tip sample forces of 50 piconewtons and below. For comparison, the approximate strength of a single covalent bond is about 1 nanonewton. The high force curve rate makes peak force tapping a viable imaging technique, capable of taking a high-resolution image in less than a minute. At the same time, it has two very practical advantages. As cantilever dynamics do not come into play, the tuning of the AFM becomes trivial, to the point where the instrument can capture optimal results on a broad range of samples in a completely self-optimizing fashion. And the force versus extension curves for each pixel can be analyzed in real time, and the obtained sample characteristics displayed as individual data channels. Another traditional drawback of AFM is the imaging speed, with a typical time of 5 to 10 minutes and more to obtain a high-resolution image. This can limit productivity. The Dimension Fast Scan uses new technology in all components of the AFM feedback loop, and especially in the AFM cantilevers, to increase the imaging rate by typically 20-fold, without impacting data quality on all types of samples. Finally, AFM have traditionally either achieved the highest resolution performance or provided large platform flexibility for large samples or multiple sample automation. The latest generation now combines the two. Let's look at these new enabling technologies in some more detail. Here we see a force versus extension curve as it is acquired for each pixel of the AFM image. The surface is on the left side of this graph. Coming from the right, the cantilever initially feels no force exerted from the sample surface. Coming close, there is a small attractive force. Under vacuum conditions, this would be the attractive regime of the Lennard-Jones potential. In ambient conditions, this attractive regime is much greater and typically caused by meniscus forces from the thin water layer present on all surfaces exposed to air. Going further, the force becomes repulsive, as the cantilever is touching the surface. When a set maximum force, the peak force, is reached, the cantilever starts retracting from the surface. In the example above, the repulsive curves into and out of the surface are different, indicating a plastic or viscoelastic component. When moving beyond the initial point of contact, the force becomes negative until the cantilever breaks free from the surface. This component represents the tip sample adhesion. The area enclosed in the curve is the energy that was dissipated during the interaction. The feedback on the peak force makes AFM imaging very intuitive, as the AFM feedback loop truly maintains a physical quantity, the maximum tip sample force. All the other components can be extracted from a real-time curve fit and displayed as separate data channels simultaneously with the topography image. An example. This figure shows an image of a polymer brush deposited on a surface. The image was impossible to obtain in tapping mode, because the tapping amplitude was either too great for the fragile structures or too small to break free from the adhesive surface. In peak force tapping, the structure could be imaged without complication. Furthermore, we can see all the different material property channels. In addition to height, we see that the droplet-shaped area containing the brush is softer, stickier, more dissipative, and more strongly deformed than the adjacent area. Another breakthrough was achieved in imaging speed. The achievable imaging speed of an AFM is set by the ability of the AFM's feedback loop to respond to the surface. Each component has a characteristic transfer function which defines how fast this individual component can be performed in the feedback loop. Taken together, these individual component transfer functions set the full system transfer function, the performance of the whole feedback loop for disturbances of different frequency. Here you see a measured full system transfer function of a pretty good standard AFM in red and the full system transfer function of the fast scan system in blue. Both start out flat for low frequencies and eventually roll off for high frequencies. However, the full system transfer function of the fast scan system rolls off at about 10 to 20 times higher frequency. So this is the improvement in imaging bandwidth that can be expected at the same image quality and the same level of force control. All academic efforts to build faster AFM have recognized the AFM probe as a key element one needs to address to increase imaging bandwidth. Concurrent development of faster AFM and affordable, high-yield commercial probes has probably been one of the main bottlenecks to the broad adoption of high-speed AFM. For most imaging modes, the first resonance frequency is the bandwidth limiting characteristic. The resonance frequency depends on the spring constant and the mass, so to increase it, one can increase the spring constant or decrease the mass. Increasing the spring constant would be at odds with our goal to increase speed while maintaining a low imaging force compatible with all types of samples. So we need to reduce the mass, and that means to make the cantilever smaller. The Bruker fast scan probes achieve an improvement in bandwidth of greater than 10x for a given application. 3 different varieties are available to cover tapping mode in air and in fluid, as well as peak force tapping. Contact mode is limited by the contact resonance, not by the first resonance of the cantilever, so it is not so critically limited by the available probes. To give you an idea what that looks like in practice, here's a real-time view of the Dimension FastScan AFM imaging a CellGuard battery separator membrane. CellGuard is created in a directional stretching process. CellGuard is a good AFM speed test sample because of the fine vertical filaments are freely suspended between the more robust horizontal elements. Therefore, one can see how the AFM tracks the surface into the gaps between the filaments and how well lateral forces by the tip are controlled. If the forces would increase at the higher imaging rate, the filaments would be deformed or destroyed. The most general situation in which the user benefits from higher imaging speed is when exploring an unknown heterogeneous sample. Most of the time is spent in understanding the different morphologies, how they relate to each other, which ones are representative of the sample, before ultimately capturing a small representative set of images for publication. In this picture of PTFE polymer, the 20 micron 16 megapixel image on the left side was taken in only 8 minutes. All the images on the right are offline zooms of this dataset. The two blue frames are the corresponding tapping phase images for two of the zoomed areas, which was also acquired simultaneously for the whole area. The image shows height sensor data, meaning the color represents true Z metrology of the topography. In this experiment, only 8 minutes of tool time were needed to capture enough of the sample surface at high resolution to later explore the data, understand the morphologies, and extract a representative set of high-resolution figures for publication. Another way to employ imaging speed is to increase throughput and gather data on more samples or at more sites on a large sample, for example a wafer. For imaging speed leading to this kind of productivity increase, it must be combined with excellent data quality, robustness, self-optimization, and programmability. We will see some examples of AFM screening on the dimension FastScan shortly. Here we'll see a movie illustrating multi-sample automation, scan speed, self-optimization, and ease of use. Some of you may recognize this movie is following the format of a popular action TV show, and this is not by chance, because at the bottom left we have a security camera. We will keep it on the instrument to show and convince you that the user is just starting the process. He has just loaded the last sample and had previously programmed just the X and Y coordinates of the stage where those samples are located. And now he goes away, and as you can watch, he is also not coming back. From now on, the instrument is on its own. The system just engaged to the first sample, and we are now at a 3 times time lapse to save you some time in watching. The real-time clock is in the bottom left corner. So this is a sample of SBS block copolymer, which is reasonably challenging for resolution, and the system self-optimized well to image it and is now withdrawing. Now watch the stage, moving to the next sample. Smart engaging, I'll tell you about that in a minute. Self-optimizing the parameters in peak force tapping and already imaging, and it is the Selgard sample. Selgard sample almost done. System is withdrawing again and moving to the next location. And now the smart engage happens, which measures the air dampening between the cantilever and the sample. So the system can come close to the sample very quickly in a matter of seconds before doing the final approach with nanometer precision. And this really enables the throughput just as much as the fast imaging. So this is another copolymer with a 25 nanometer line width pattern, withdrawing again. Moving to the next sample. Smart engaging. Here now you can see some extra structure in the image next to the cantilever, because the camera's purpose is to monitor the cantilever just as much as to see and to navigate the sample. The earlier samples only had nanoscale structure, so we couldn't see anything. The sample has sufficient macroscopic structure to watch and see how it is being scanned. And this polymer sample is now complete. Moving to the next sample. Self-optimizing. auto-scaling the data for display, and we see this is the etched mica again. So this is hugely different in mechanical properties as well as demand on resolution with the half nanometer steps, and the system self adapts to that well and displays it in good quality. And finally moving to another sample, and we see some macroscopic structure here again. It looks like fibers. It's self-optimizing and auto-scaling, and we identify this as collagen fibrils, actually from a rat tail. You see the characteristic 67 nanometer debanding in the collagen fibril. And from now on, we are back to real time. So you now see the actual speed at which the data is being obtained in the system right now. And 10 minutes are up, and we have imaged 6 samples in 10 minutes, hugely different samples, all fully automatic with no user present. After having convinced ourselves that the most current AFMs have evolved to be quite comparable to, for example, current SEM with regard to imaging speeds, usability and richness of information, we will now take a look at how the information provided by AFM is unique and complementary to other techniques. The AFM provides high resolution on non-conductive samples, while with true 3D metrology and additional nanoscale material and, if desired, electrical properties. The required prep is often minimal, as the AFM typically looks at native sample surfaces, often in their native chemical environment. Responses to changes of this environment can be directly observed. Imaging in fluid also enables imaging biological systems up to live cells. The data storage industry is driving to ever higher hard drive capacities. Hard drive media consists of a fast-spinning disk with a thin magnetizable coating. A read-write head is flying a few nanometers over the disk, stabilized at the height by the aerodynamic forces of the spinning disk. As data densities increase, the gap size between head and disk decreases, and the flatness of the magnetic media becomes ever more critical. The disk substrates now typically consist of glass, and the surface flatness is achieved through a chemomechanical polishing process. To optimize the process parameters and to verify the quality of the product, samples are taken from the batches and nanometer roughness measurements are performed by AFM. Taking measurements on more samples and at more sites provides more data context. More data context increases confidence in the data. Repeat measurements eliminate the possibility of site-to-site measurement error due to tip abrasion. In this example, a 512 by 512 pixel AFM image was taken for each roughness measurement. Roughness was measured at 8 different angles and at 3 different radii of the disk media substrate. The measurement was repeated 4 times to verify repeatability. The complete dataset of 96 measurements shown above was taken in 2 hours. Now to the analysis. The area roughness in nanometer of all acquired images was determined after image flattening and exported to an Excel table using the batch processing in the nanoscope analysis software. The top graph shows the raw data from the 4 runs. Below are data averages, binning radii, and runs for a plot versus angle, angles and runs for a plot versus radius, and run averages to check for tip wear. Generally, the measurements from each run follow the same local trends and fall within 5% of each other. Tip wear would manifest itself by a decrease of roughness from run to run. There is a variation of run roughness average of less than 2%, but a decreasing trend cannot be observed. The roughness versus angle shows a local peak and a valley on the opposite side of the disk. Further investigation could be interesting here. Increased local roughness could be due to sample contamination. For reduced roughness, this is less likely. However, the effect is within the standard deviation of the measurement and doesn't show trends for example a sinusoidal. A slight linear roughness variation as a function of disk radius seems more consistent in this data and could be considered for the polishing process. This data requires the combination of excellent Z-noise performance and large sample automation as a minimum. The fast scanning capability moves the measurement time from an impractical 20 to 40 hours to a more reasonable 2 hours. We will now look at some examples of imaging nanoscale processes. This is enabled by the AFM's ability to image native sample surfaces at high resolution while controlling the physical and chemical sample environment. This movie shows the de-wetting of a 5 nanometer thick polystyrene film from a hydrophobic silicon surface. The process is heat-driven. The experiment was performed using the dimension fast scan in conjunction with the dimension heater-cooler. The data was recorded with 512 lines at 22 seconds per frame. Initially, the sample is heated to 55 degrees C. Now, hole formation can be observed, one at a time, consistent with thermal nucleation. When the de-wetting process slows down, the temperature is increased to 65 degrees C and finally 75 degrees C. This experiment was performed in collaboration with a group of Karin Jacobs at the Universität des Saarlandes. To study dynamic processes, it is, in addition to the imaging speed, also important to be able to trigger and control them. The open tip scanning design makes it fundamentally easy to accommodate heaters, chambers, and other devices that provide process control to the sample. Bacteriorhodopsin is found in archaea, the most notable being halobacteria. Bacteriorhodopsin acts as a light-driven transmembrane protein pump, converting light into chemical energy for the organism. Purple membrane is a two-dimensional protein crystal formed by the membrane protein Bacteriorhodopsin. BR consists of 3 identical protein chains, arranged by a 120-degree rotation. High-resolution imaging of membrane protein crystals like purple membrane is one of the most taxing samples for the performance of an AFM with regard to resolution and force control in fluid. The AFM image on the right from a 2008 review represents the pinnacle of a decade of membrane protein research by AFM with regard to its quality and resolution. Here we see a movie of 3 larger purple membrane patches, loosely adhered and diffusing on a mica substrate in buffer solution. The movie was taken at a rate of 22 seconds per frame and consists of 75 frames in total. The imaging mode used was peak force tapping. Let's take a closer look at the movie. We see 3 purple membrane patches, one on the left, one on the right, and one mostly overlapping on top of the right one. This one is hanging down like a waterfall, and in the beginning of the movie, a piece of it separates and self-integrates into the left patch. Shortly thereafter, another piece self-integrates into the right patch at the top, as we'll see in a moment. Here is the piece in the circle. It separates and now integrates, and this piece also integrates into the other patch. Now we're adding more protein to the sample solution, and we see how this protein is coming down as we have more material on top of the patches. What is interesting is that the pieces now are self-integrating, while the pieces that come through the solution don't appear to be self-integrating. Clearly the self-integration happens over the edge of the patch, but not over the patch surface. However, you see that some of the material that was added to the solution stays on the surface and is actually highly mobile. Especially now on the right-hand side, you can see a couple of Bacteriorhodopsin small patches moving around on the big patch. We can see that a lot of insights about the behavior of nanoscale sample, which is not apparent in still images, can be obtained from a time sequence. Here again, the data we saw earlier in the presentation. The acquired Bacteriorhodopsin data after background subtraction. The 2D FET reflects the hexagonal symmetry of the protein crystal. The spectrum shows good symmetry, low distortion. The inner maxima represent the unit cell, the outer ones the trimeric substructure, as expected from the X-ray crystallography data. The central pore is well resolved. This data was taken with peak force tapping with a peak force tapping amplitude of only 2 nanometers. After this brief update of how far AFM has come with regard to ease of use, performance, speed, and data interpretation, we will now look at some examples where both an AFM and an electron microscopy technique was used on the sample type, on the same sample type or application. This will give us a better feel for how the techniques are similar and how they are different or complementary. This first example shows an SEM image of a diatom mineralized shell or frustral. The SEM provides a good sharp overview due to its almost infinite depth of field. However, this is an empty shell. The AFM shows the sieve structure on top of the diatom, in this case a live diatom imaged in water. The peak force quantitative nanomechanics mode shows the topography as well as the modulus, adhesion and deformation mechanical data. We can clearly see the differences between the hard mineral shell and the soft, adhesive and deformable biological matter inside. Earlier AFM studies have used similar techniques to study the adhesive the diatom produces to hold onto the surfaces, such as rocks or the aquarium window. In the growing field of biological pharmaceutics, therapeutic antibodies are mass-produced to serve as a drug substance. It can happen that some of the antibodies from complexes, for example dimers, and some of the form complexes, for example dimers, and some of these complexes can be toxic. It is therefore important to produce and formulate these antibody solutions in such a way that complex formation, especially of toxic species, is minimized. This can be addressed through nanoscale imaging and subsequent particle analysis, as shown in this very recent papers from authors at Hoffmann-La Roche and the University of Basel. They use TEM for their study and again the computational methods to analyze the images, reconstruct high-quality representations of the complex species, and assignment of individual particles to these species for statistics are well developed for TEM applications. On the right we see monoclonal antibodies imaged by AFM at cryo conditions for optimal resolution. It can be seen that many of the antibodies imaged by AFM already show comparable quality to the average representations obtained with TEM. It seems likely that, given a similar attention to sample preparation, data analysis, etc., AFM would be a similarly useful tool for formulation development and quality control of protein solution-based drugs. Here is a classic example of TEM imaging performance. As light microscopy gave us the first glance at bacteria, electron microscopy gave us the first pictures of virus particles. Electron microscopy is today a standard tool to identify rare virus types for which biochemical tests are not always available. On the left we see TEM image of a herpes type 1 virus. On the right, the same virus by AFM. This example shows nicely the TEM's emphasizing of membrane structures vertical to the image plane, and the AFM's emphasis on the three-dimensional structure of the same object. The resolution appears comparable by eye. The TEM has a technical lateral resolution that can be higher than the AFM's. However, this assumes that the sample structures are sufficiently well prepared during the often invasive TEM prep, and that the contrasting agent finds enough binding sites to bring out the resolution. It would be interesting to compare TEM and AFM of virus particles in routine virus particle ID. Here we see a bone fracture surface, imaged by different techniques. This is interesting. Looking at the fracture surface shows the bone constituents that failed during the fracture. Osteoporosis, for example, cannot be explained by the loss of mineral alone. A few percent of bone mineral loss are accompanied by 80 to 90% of bone strength, loss of bone strength. In this paper by Fandner et al., we investigated the constituents that serve as a glue between the mineral parts and the collagen parts, and their organization on the nanoscale. The first image was taken by SEM, the others by AFM. Each technique is capable of revealing the collagen debending, left and middle, and some apparent coating on the left and the right image. This AFM experiment brings the pieces together. First, the fresh fracture surface was imaged, as in the A area. Then the sample was briefly exposed to dilute EDTA, a chelator that selectively removes hydroxyapatite mineral. Now the same location was imaged again, and the removal of the mineral revealed the underlying collagen fibrils, as seen in the inset B. This experiment would not be possible after conductive coating of the untreated bone, and even in the case of ESEM, would be infinitely harder to do with the required treatment of the sample outside the instrument and re-imaging of the exact same location later. Polymers are a large application area for both electron microscopy and AFM. This example from TU Graz shows both the strengths and the weaknesses of TEM and AFM on an interesting sample in comparison. The sample consists of two different polymers, with an embedded layer of silver nanoparticles in between. The TEM clearly shows the embedded nanoparticles, but provides no differentiation between the polymers. The AFM phase image shows the different morphology of the two polymers, but only reveals the silver particles exposed on the surface. Taken together, the two techniques provide a complete picture of the sample. Transmission electron microscopy has a 60-year head start on AFM in terms of methods development. What can AFM learn from this wellspring of experience? It is likely that some of the imaging performance reached by electron microscopy is owed to the sample preparation and contrasting techniques originally developed for electron microscopy, but would benefit and enrich AFM applications if applied there. The classic methodology for imaging tissue samples by TEM is a fixation step followed by a substitution of the water with a non-volatile cuttable medium, followed by sectioning. It is important to note here that the resolution of any microscopy technique on a heavily prepped sample is only as good as the used prep technique's ability to preserve small structures. The latest incarnation of this prep uses high-pressure vitrification, where the sample is cooled to sub-zero temperatures while kept at high pressure to prevent the water from freezing. Once the pressure is released, all water instantly transitions to the solid phase. Since no heat transfer is involved in the second step, the process is fast. This prevents crystalline ice formation, which in turn preserves the membrane structures on the nanoscale. In the next step, the water is substituted with a resin while in the frozen state. Finally, the sample is sectioned with an ultramicrotome. For AFM, the quality of the diamond knife is essential to avoid imaging of knife-generated topography later. Special AFM ultramicrotome knives are commercially available now. For TEM, a thin microtome section is imaged after adding a heavy element stain, for example uranyl acetate, which selectively binds to certain structures in the tissue to generate contrast. For AFM, the sectioned block face is imaged. When imaging the last section slice with TEM and the block face with AFM, a complementary dataset can be generated. Below some examples of such complementary datasets from different tissues by Naderta Matzko at Technical University of Graz, who pioneered this technique. This is a cross-sectional image of an ant antenna, high-pressure vitrified and free-substituted with a resin. The antennae serve the ant as an olfactory organ. The rigid chitin outer structure has many openings to expose the sensory cells to the environment. This scan is an overview with 25 megapixel resolution, imaged with a dimension fast scan AFM in peak force tapping mode at a rate of 8,000 pixels per second. The image has a scan size of 30 microns and a pixel resolution of 6 nanometers. All subsequent analyses we will see of the sample are data zooms from this one dataset. This overview shows one practical advantage of AFM when compared with TEM, are the relatively large overview scans that are easily possible. While many TEM instruments would allow similar scan sizes, the need to use a grid and position the section sample region of interest in one of the grid holes puts some practical constraints on large TEM scans of tissue sections. Here we see a data zoom of the central region of the previous 25 megapixel image. Appropriate flattening starts to reveal detailed structures in the tissue. When we further compare this region imaged by AFM and the image of an adjacent TEM microscope slice, we see that each shows detail of the cell organelles. The TEM emphasizes structures that are vertical to the image plane, because the electron beam transitions more uranyl acetate stained material on a vertical structure. This leads to a pronounced 2D appearance. The AFM images the surface of structures, therefore it emphasizes the three-dimensionality of compartments, etc. In addition, the TEM stain has an affinity for the membranes, while AFM emphasizes physical size. In membranes, the membrane proteins are dimensionally dominant. Both images are beautiful and provide two different views and therefore a better foundation for interpretation of the same or similar structures. Antimicrobial peptides are naturally occurring small molecular weight molecules that are part of the innate immune response of all classes of life. With the increasing problem of antibiotic-resistant bacteria and the selectivity of antimicrobial peptides for microbial cells, AMPs are now being evaluated as potential therapeutic agents. AMPs permeabilize the microbial cell membrane through membrane disruption and pore formation, mainly through three established different models: the barrel stave model, the toroidal pore model, and the carpet model. The barrel stave model just forms stable pores in the membrane, while the latter two finally lead to micellization of the cell membrane. We will focus here on a study done with the antimicrobial peptide CM15, which is supposed to follow the toroidal pore model. This work was inspired by a couple of AFM projects, including one high-speed study on whole bacteria by Georg Pfandner at the EPFL in Lausanne. Three live E. coli bacteria imaged by fast scan in fluid can be seen in the movie, taken at 18 seconds per frame with 150 kHz, 0.3 nanometer, 8 micron wide cantilever with EBD tip from NanoTools in Munich. These cantilevers are fairly pricey, approximately 200 euros per piece. But the data shows that running experimental cantilevers smaller than our broadband probes is definitely possible with a fast scan. The data shown is phase, which contrasts the changes more clearly. The cells were exposed to 10 microgram per milliliter CM15, which is known to be the minimum effective dose. A number of studies by different groups using AFM to investigate the effects of various AMPs on live bacteria cells have observed similar dramatic changes to cell morphology, with increase in roughness and corrugation of the cell surface. This slide and the next probably show the most exciting data in the study. On the right is a high-resolution AFM scan of the bacteria surface showing ordered structure consistent with the bacterial S layer, as seen in the TEM image by Dupré et al. On the left. The AFM inset shows the whole bacterial cell and the area on which the high-resolution scan was taken. Both techniques show comparable ability to resolve the features. However, the AFM sample is a whole E. coli bacterial cell in fluid, and it is still alive. Now watch the time series of the bacteria surface at this resolution. The movie consists of 45 frames, each taken within only 8 seconds. The data shows the roughening and disruption of the S layer in response to the CM15. This is consistent with the toroidal pore model, from which we expect eventual micellization of the cell membrane. The delay between CM15 exposure and onset of cell surface roughness increase can be seen in the synchronized graph on top. This control experiment shows the bacteria before and after taking the previously shown high-resolution data. The changes occurred homogeneously across the entire bacteria surface, not just the 300 nanometer scan window. This shows that the changes were induced by the CM15 and were neither induced nor significantly accelerated by the AFM imaging process. The data also permits another observation. The CM15 exposed cell is smaller. This is in agreement with SEM observations by Hartmann et al. However, SEM observation requires fixation, while AFM permits nanometer precision size measurement on a live cell before and after exposure. The SEM study raised the possibility that CM15 exposed cells are inhibited in their growth. The AFM data clearly shows that a live cell exposed to CM15 undergoes shrinkage. Finally, let's review again the examples we just saw. For the recent advances which make the AFM more suitable for environments used to the productivity of electron microscopy. Scan Assist, the self-optimization of AFM imaging based on peak force tapping, was shown in the productivity example of the 6 samples in 10 minutes and on the very challenging high-resolution example of the Bacteriorhodopsin lattice. Mechanical material property contrast by peak force quantitative nanomechanical mapping was illustrated on polymer brushes. The combination of large sample and multi-sample automation with highest AFM resolution, stability and noise performance was verified in the mica lattice image shown in the beginning and critical for the 6 sample example, the media substrate roughness screen. Almost all examples shown in this presentation took advantage of the greatly improved imaging speed enabled by the Dimension FastScan system. Thank you for your attention.
Thank you very much, Johannes. And we will now open a portion of the presentation up for questions. There's a number of questions that have already come in, and I will try to group those together and answer them, and have Johannes answer them so we can get through the most of them. I also noticed that a number of you have your hands up. There's a feature within the software which lets you raise your hand. If you have that so you can ask a question, I would appreciate it if you would instead type your question directly into the questions box, and we will review it from there. So, Johannes, there are a number of questions on sort of moving through the presentation from the beginning on Scan Assist, particularly, you know, what happens with the Smart Engage, and will this work on samples of different Z heights? So could you talk about that, and also just review how Scan Assist works?
Okay. So fundamentally, the way Scan Assist works as compared to other imaging modes, Scan Assist is a feature that uses the peak force tapping mode. And the peak force tapping mode is different from contact mode imaging or tapping mode imaging. In contact mode imaging, the cantilever is in constant contact with the sample and basically slides over the surface of the sample. In tapping mode, the cantilever is freely oscillating at its resonance frequency, and what is measured to follow the surface is actually the dampening effect of the surface onto the cantilever. In peak force tapping, we are taking individual force curves of the sample, so the difference to tapping mode is that the frequency of movement of the cantilever is way below the cantilever's resonance. So for example, where in tapping mode you would maybe, to get 1 pixel, you would oscillate over that pixel area 100 times. In peak force tapping, we take just 1 force curve, approaching the surface and retracting from the surface. So Scan Assist can use this because if you look at what is the biggest complication in automatically tuning up an AFM, in contact mode the biggest problem is that there is an unknown force offset to the cantilever, which in the beginning gets set to 0 when the AFM gets set up and the laser gets set to the center of the detector. But this can change over time, and there's no way to know while you are imaging that is happening, because you're always in contact, so there's no comparison. In tapping mode, on the other hand, the biggest complication is that you are using the free oscillation of the cantilever, and therefore all the physics associated with harmonic oscillators, dampened harmonic oscillators, bifurcations, in some situations even chaos and so on, are part of your imaging process, and it takes an experienced operator to know what to do in order to maintain, set up and maintain stable imaging conditions, especially on more challenging samples where there is a lot of different interaction with the cantilever, such as adhesion, for example. In peak force tapping, when the cantilever starts approaching the surface, it is initially in free air, so we have a reference for what zero force looks like for each pixel. Then it comes into contact with the surface, and we know exactly what the force between the cantilever and the surface is, because it's basically the delta between that initial deflection when you're off the surface and the deflection when you're on the surface. And we know, because it's a common characteristic of force curves, that they can have a larger or smaller adhesive component, and after that it's repulsive. And so basically we look for that adhesive regime to be over and the repulsive regime to start, and then we just wait for that peak force, and we feed back on that. So this is highly robust because the cantilever physics basically don't, except for the spring constant, of course, the cantilever physics basically don't play a role, and therefore it is a much more straightforward, much simpler way to explore the surface, and it is much easier to design an AFM that tunes up to that automatically. Speaking of the topography and the different samples, so I think I answered a piece of that. Basically, the sample differences that matter to the AFM on the one hand are the mechanical properties, and if they become extreme, for example, a thick water layer that can be much more robustly handled in a force curve than in a dynamic imaging mode. With a free cantilever oscillation and so on. Concerning the Z heights, within the range of the AFM, we can basically just auto-adjust the data scale of the A to D converters to be optimally suited for that sample after a few lines of imaging. But the other optimization that's going on is definitely also the scan rate and the optimal interaction force for that sample, which can basically be minimized without losing the capability to track the sample. And we saw those forces can be quite low in the 50 piconewton range. Of course, comparing SEM with AFM, there is one big difference. The SEM is basically the perfect the perfect photo camera in the sense that it has almost infinite depth of focus, where the AFM you are physically basically limited to the length of the cantilever tip as a maximum, because after that the cantilever itself will come in contact with the surface. So there are certain classes of samples. If you want to take a photograph of a whole ant, for example, then SEM is your technique. But as soon as you come into roughnesses that are in the range of a couple of microns, the AFM comes into view.
So maybe that's good. A good segue into a couple of these questions, and I know we're running a little late. I'd like to hit maybe one or two more. So on sample prep, the question is, how do you image a tissue sample in the AFM? Essentially, how do you prepare it for scanning?
Okay, so I showed these examples, but I think there are somewhat groundbreaking work actually coming from the TU Graz looking at these sections. Typically, the AFM at the moment is used for biological material up to eukaryotic cells, individual cells. For multicellular tissues, this is definitely an interesting area, but it is also challenging as it is to any microscopy technique, and at some point you cross over into the optical microscopy regime. So in this particular case, the tissue was prepared in the same fashion as it was for TEM. Basically, this was an example of using a classic TEM prep for AFM, where the sample gets high pressure frozen, and then the water got pre-substituted with the resin, and then sectioning was performed, and actually the last section was used with the TEM while the remaining block phase was used for the AFM. And interestingly, even though you apply basically a flat cut to that surface, you already have a few nanometers of topography, which is the image I showed. You can further enhance that contrast, as I learned working with a group that pioneered this technique. You can further enhance this contrast by a short brief exposure to a clean solvent, like for example clean ethanol. And basically what happens there is that part of the epoxy is polymerized, but other parts are not, and are easily dissolved. And at that point, immediately the contrast between a material containing tissue and material just containing epoxy is exaggerated and goes into the tens or even hundreds of nanometers. So that contrast can be dialed in by the time of exposure there.
Okay. So I think we're running a little bit over, and so maybe I'll make this the last question, and also invite everybody to submit your final questions into the questions dialog box. And while we won't be able to answer them online today, we will follow up with the answer to your questions on email. So this is keeping with the biological imaging, but more on the Q&M side than the sample prep. Does the AFM adhesion map, is it dependent on the topography of the sample? And I think that this came up during the image of the diatoms.
Okay. So if you, I think the short answer is yes. From a certain amount of roughness on, there is definitely a contribution of that, and one has to be aware of that. And where that would come from is basically a change of the contact area between the cantilever tip and the sample. So the cantilever tip has a certain tip angle, and as soon as you have components that are even approaching that, you start having some level of side contact, and so you exaggerate your stiffness and probably some of the other components simply by the fact that that you're not imaging with a hemisphere anymore, but you're imaging with start imaging with a cone. So one has to be aware of that when making quantitative claims, definitely. And actually, maybe this is where the question comes from. If you look very closely at the diatom image, what you see is the frustral, and it is hard, and then low adhesion. And if you then look at the individual holes, they are soft and high adhesion, but in the stiffness one, there's actually a small dip in the center of the holes where there seems to be high stiffness again. And I would actually guess that that is probably a sidewall effect happening in the deepest region of that hole. So one has to be aware of it, but like with all techniques, you have to know the limits of the technique and then exploit it within those constraints. And where the sample is sufficiently flat, which can be actually fairly rough, just not over 45 degrees or something, then you get very good data.
Okay, thank you very much. With that, I will conclude this webinar. Please do remember to take the exit survey